Hi, everyone. My name is Parzad. I work for the Harvard Brain Science Initiative, and we're really excited to have the first basic science event in our 101 series, Synapse Elimination 101, featuring Drs. Hisashi Umemori and Beth Stevens. Both of them are associate professors at Harvard Medical School and the F.M. Kirby Neurobiology Center at Boston Children's Hospital. They have labs near each other and work together. And yeah, we're very excited to have them. So just a few words about each of them. Dr. Umemori's lab studies molecules and mechanisms that underlie the formation of synaptic circuits in the brain. And also they are interested in how these circuits and the wiring may go wrong in brain disorders. So Dr. Umemori first started receiving an MD at the University of Tokyo, where he became interested in psychiatric diseases and realized that there was also that he might want to research the basis more doing basic science research. So then he did his PhD, also at the University of Tokyo, and looked at molecular mechanisms of myelination and synaptic plasticity. And then that interest in synapse development brought him to Josh Staines's lab at WashU St. Louis, and then eventually Harvard. And Dr. Umemori was a faculty member at the University of Michigan and then came to Harvard in 2013. And he's received numerous awards. And Dr. Stevens, in addition to her appointment that I mentioned at Boston Children's, is an institute member at the Broad Institute and the Stanley Center for Psychiatric Disease Research at the Broad. She's also a Howard Hughes Medical Institute investigator and a member of the National Academy of Medicine. So her lab is interested in neuroimmune interactions in the brain and understanding how such interactions might sculpt synapses and how this can be important during both healthy brain development and brain disease, as she'll talk about today. And her team is famous for their discovery about how microglial cells, which are the immune cells of the brain, can prune neural connections. And how that can happen in response to signals from the classical complement cascade, which was previously known a lot about that in the immune system, but not at all in the nervous system. So Dr. Stevens did her postdoctoral training with Ben Barres at Stanford. And prior to that, she did her graduate work at NIH and the University of Maryland. And she's also received many awards, including the MacArthur Genius Award in 2015. So yeah, we're very excited to have both of you here today. And in terms of format, Dr. Umemori will speak for about 25 minutes, and then we welcome any specific questions for his talk for about 5 minutes. You can type them in the chat box or raise your hand, and then Dr. Stevens will speak, and then after that, again, specific questions for her talk, and then we'll have about a half hour for a broad general discussion. So yeah, I think we'll get started.
Good. Okay, so thanks, Parizad, for the introduction. So let me share my slides. So here, do you see my first slide?
Yes.
Good. Good. So today's topic is synapse elimination. So first, what is synapse elimination? There is an analogy or analogous theory called the buffalo theory. I don't know how many people have heard of this, but this is from a TV show called Cheers. I think it's in the 80s, but there is a character called Cliff Clavin, who is a kind of wise man, knows about many things and talk about many theories at the bar while drinking. And this theory is one of them, the buffalo theory, which goes like this way. A herd of buffalo can move only as fast as the slowest buffalo. When the herd is hunted, it is the slowest and weakest ones at the back that are killed first. This natural selection is good for the herd as a whole because the general speed and health of the whole group keeps improving by the regular killing of the weakest members. And this theory goes on like this way. In much the same way, the human brain can only operate as fast as the slowest brain cells. Excessive intake of alcohol, we all know, kills brain cells, but naturally it attacks the slowest and weakest brain cells first. In this way, regular consumption of beer eliminates the weaker brain cells, making the brain a faster and more efficient machine. That's why you always feel smarter after a few beers. So this theory is a concrete proof that getting drunk is good for your intelligence. And this theory finally goes on to talk about synapse elimination or synapse refinement, okay? Synaptic circuits in our brains, like the neurons from the buffalo theory, refine themselves naturally through intercellular competition without alcohol. So that's the idea about synapse elimination, and you can read the connection between the buffalo theory and synapse elimination on this website on IO9. But this is the topic we'll be talking about today. So synapse elimination or synapse refinement, which is first characterized in the sensory motor systems, specifically at the NMJ and the visual system. In both systems, initially intermingled and excess synapses would be refined to form more sophisticated connections. Like here at NMJ, there is a one-to-one connection between the motor nerve and the muscles. And in the case of the visual system, you see more segregated targets from the right and left eyes. In the visual system, the first observation of refinement was done by Hubel and Wiesel, who are both at Harvard, who also knows about the effect of alcohol apparently, but who focused on a problem during the treatment for amblyopia and strabismus. An effective treatment of these diseases is to patch the dominant good eye to force the child to use the non-dominant eye like this way. But the problem is the child may have reduced vision in the good covered eye. So they wanted to know why this happens by examining the connections from the right and left eyes to the thalamus to the cortex layer 4. And this is what they found. So they found that the connections from the left and right eyes to the cortex are initially intermingled like this way, but then eventually segregated to form dominance columns. Like these are from the right eyes and these are from the left eyes. This segregation happens during development in the case of a monkey between the birth and 4-week-old. Initially intermingled connections will become more segregated like this way. And when you close one eye at the time of birth, this will happen. An open eye will basically occupy the entire area in the cortex, and a closed eye loses its connection to the cortex. And that is why you lose the vision of this covered eye. For this phenomenon to happen, there is a window. For example, if you close one eye at 6-week-old, then nothing happens. And younger, the worse, meaning there is a critical period for this ocular dominance formation. So ocular dominance column formation is essentially input dependent, and there is a critical period. Like critical period is kind of studied by people like Takao here at Harvard as well. At the neuromuscular junctions, synapse refinement is also observed here by Jeff Litman and Josh Sainz, also both at Harvard. This is an example of imaging. Initially, you see there are two motor nerves innervating to the muscle like this way, but soon one motor nerve is eliminated to form an one-to-one connections here. This synapse elimination is very robust. At the NMJ, so here at E18 in mouse, this motor axon innervates around 80% of the NMJ, like this way. But by P13, you see that this motor axon only innervates around 4% of the muscle fibers, meaning synapse elimination is really a robust process. And synapse refinement is also observed in human brains. And in this case, it's more like projection refinement or refinement of processes. But here in the human brains, you see an increase in the processes from newborn to 2-year-old. Then you see a decrease This decrease continues until 15 to 20-year-old. And this is also observed by MRI studies. Like here, by examining the density of gray matter, you see a decrease from 5-year-old to 20-year-old, partly due to a decrease in the processes of neurons, meaning synapse refinement is a robust and long process. And that, why is that important? Okay. So because defects in synapse refinement are linked to various disorders. So this is the normal development of synapses. You see synapses are formed, followed by synapse elimination, and then maintained in the normal subject. In the case of autism, you see more synapses are formed and fewer synapses are eliminated here. In the case of schizophrenia, more synapses are eliminated here. And as a result, you see more synapses or spines in autistic patients and fewer spines in schizophrenic patients. Similar mechanisms may be also involved in synaptic loss in the adult in neurodegenerative disorders like Alzheimer's. And this topic will be discussed by Beth Stevens later. So synapse refinement. So what I want to discuss today is how the neurons decide to refine synaptic connections. How do they know which ones to keep, which ones to eliminate? So we've been studying synapse refinement in the brain, such as in the hippocampus and the cortex. And today I will show you an example of synapse refinement of the colossal connections. So as you know, colossal connections are the bundle connecting the right and left hemisphere. That's the largest bundle to connect or transmit information between two hemispheres. And defects are kind of connected to autism and schizophrenia. And by labeling neurons on one hemisphere, you can clearly see colossal bundles, colossal axons like this way. So we first examined the development of colossal connections. And for this, we labeled one side of cortical neurons by introducing EGFP by inhibitory electroporation to label neurons here to examine colossal axons like this way. And here's the development in mouse from P0 to P30. And what you can see are like these are colossal axons, and these are high magnification. And you can see an increase from P0 to P5 followed by a decrease as quantified here. So this means from P0 to P5, synapses are being formed, followed by synapse elimination from P5 to P15 of colossal connections. We then wanted to know whether this is activity dependent or whether we are keeping good ones and eliminating bad ones, like in the buffalo theory. And for this, we introduced tetanus toxin light chain, which is to inhibit neurotransmitter release in addition to EGFP into one side of the neurons. Here, we introduced TTLC only into around 30% of neurons, creating this kind of situation, meaning around 30% of neurons will be inactivated here, but the remaining 70% of neurons will be still active here. And in this case, these inactivated connections are formed here, but then eliminated, which is quantified here. So initial formation is not affected by inactivation of neurons, but then inactivated connections are all eliminated by P15. So this means indeed inactive connections are eliminated. In this case, these connections can be eliminated because we still have good connections here, right? We then wanted to know what will happen if we inactivate all connections like this way, okay? And for this, what we did was to, after introducing tetanus toxin and EGFP into a subset of neurons, applied tetrodotoxin to globally prevent neuronal firing to create this kind of situation In this case, all connections are inactivated. In this case, these inactive connections are maintained as opposed to eliminated. Okay, so here's a summary so far. So in this case, if like connections are active, they're maintained. In this case, these connections are eliminated. But if they're all inactive, these connections are maintained. This means inactive connections are eliminated only when there are other active connections. In other words, there will be a competition, and active ones are the ones to send signals to kick inactive ones out. Okay? And as a result, there will be a signal within the inactive connections that is detecting this punishment signal to drive the elimination of inactive connections. At the same time, there will be a signal to stabilize these active connections. So then the next question is whether indeed these signals exist, and if so, what they are. I'll first talk about this elimination signal. Okay? So this elimination signal should be activated within the inactive connections in response to this go signal. Okay? So we performed various screens to identify elimination signals, and we identified several molecules, and I'll introduce you one of them, which is a tyrosine kinase called JAK2. This is a tyrosine kinase. When this is activated, this is phosphorylated, meaning phospho-JAK2 means active JAK2. JAK2 in active neurons, which is shown here, is not activated. However, when neurons are inactivated, you see JAK2 becomes activated. But this activation requires other active neurons, because when we applied TTX to now globally suppress the neural activity, then JAK2 is no longer activated within inactive neurons. Okay? So this means JAK2 is activated in inactive connections in response to signals from active synapses. Okay? So this serves as a kind of sensor of the punishment signal within the inactive connections. And indeed, JAK2 is necessary to drive the elimination of inactive connections, which is shown here. So here, in addition to tetanus toxin light chain, we introduced a kinase dead form of JAK2. And you see that inactive connections are not eliminated. Okay? So these two are controls. And conversely, just by activating JAK2, here, we are able to drive the elimination of colossal connections. So far, these are axons or connections or projections. So we can examine functional synapse refinement by performing electrophysiology, so which is shown here. So here, we stimulate the colossal bundle and record from a postsynaptic neuron located here in the cortex. And we use two types of stimulations here. One is a single fiber stimulation, which is to elicit the smallest possible response, assuming that is induced by a single fiber. And second one is the maximal stimulation. This is to basically stimulate all the inputs to our neuron. Okay, so roughly speaking, maximum response divided by the single fiber response would give us an estimate of input number that a neuron receives. Okay, and here's an example of recording during development. Single fiber amplitude increases from P5 to P15, and that's the max amplitude. And if we calculate the input number, here's the time course during development. From P5 to P10, you see an increase indicating synapses are being formed, followed by a decrease from P10 to P15, indicating synapses are indeed eliminated during this time. Okay? So we then examined the role of JAK2 during this time. First, we activated JAK2. For this, we knocked down SOC3, which is an endogenous suppressor of JAK2. So by knocking down this molecule, JAK2 is activated from the presynaptic neuron. And these are the recording results. Before synapse refinement, no changes observed, meaning synapse formation is not influenced by JAK2 activation. But here, after synapse elimination, you see fewer input, meaning more synapses are eliminated. So when JAK2 is activated, more synapses are eliminated. Conversely, if we inactivate JAK2 within the presynaptic neuron, then you see more synapses maintained like this way. So altogether, this is what's happening. During synapse refinement, there is a competition between active and inactive connections, and active ones are the ones to send signals to inactive connections. And when this was received within the inactive synapses, elimination signals such as JAK2 is activated. And once activated, this will drive the elimination of inactive synapses. Now, let's look at what's happening at active synapses. I said that there will be a punishment signal from active synapses to attack inactive ones to drive the elimination. But this punishment signal should naturally attack other active synapses that should be maintained. So this suggests that there should be a protective signal or stabilizing signal to keep active connections. It is really true. Is this really existing? So we again searched for stabilizing signals, and we identified a candidate molecule, which is called SERP-alpha, or signal regulated protein alpha. This is a transmembrane protein located on the postsynaptic membrane, but this molecule extracellular domain is cleaved in response to synaptic activity, meaning if synapse is active, and only when this synapse is active, this portion is cleaved. And once cleaved, this portion goes back to the presynaptic terminal, bind to a receptor called CD47 to promote presynaptic maturation, meaning this molecule, SERP-alpha, is cleaved by the activity to drive presynaptic maturation. So we thought this could be a candidate molecule because this is cleaved only at active synapses to protect active synapses from elimination, like this way. And indeed, when SERP-alpha is knocked down, we see more synapse elimination. So fewer input numbers, meaning more synapse elimination at the end of synapse refinement. And when we applied SERP-alpha to neurons, it inhibited JAK2 activation, which is shown here. So here's the control, you see phospho-JAK2, but you see like by applying SERP-alpha, activation of JAK2 is suppressed. And finally, when we activated JAK2 in presynaptic neurons and applied SERP-alpha extracellular domain from the postsynaptic neuron, like this way, and this is the kind of how it's done. We were able to rescue JAK2-driven synapse elimination, which is shown here. So here, SOC3 knockout, JAK2 is activated. When JAK2 is activated, you see more synapses are eliminated. But this JAK2-driven synapse elimination is completely rescued by expressing SERP-alpha, suggesting that SERP-alpha serves as a protective signal, as shown here. So here, there is a punishment signal at inactive synapses. JAK2 is activated to drive the elimination. However, at active synapses, SERP-alpha serves as a protective signal. Okay. Now, so this is the final slide to summarize what I talked to you today, and that's a model. So when during synapse refinement, there is a competition and there is a signal from active neurons, the go signal. If this is an inactive synapse, this will activate elimination signals such as JAK2. And this, once this is activated, this will drive the elimination of this inactive synapse by eventually recruiting microglia to remove the inactive synapses. So this part will be discussed by Beth Stevens next. And if this synapse is active, now there is a local feedback, stay, or protective signal, such as SERP-alpha, which will inactivate elimination signals to keep this active synapse. So during synapse refinement, the brain is calculating those signals to determine which ones to keep, which ones to eliminate, so that the brain can establish the most efficient and functional synaptic connections, even without alcohol. So of course, there are many questions still remaining to answer. So that's stay. Like, for example, what are the nature of this go signal, right? So there is a long-standing neurotrophic hypothesis, and I still think that's a possibility, but we still don't know the actual nature of this go signal. Second, and post. So today I focused my talk on the presynaptic side, but postsynaptic cells, of course, involved, and studies like Mike Greenberg and colleagues also suggested that transcription factors like MF2 are involved in synapse refinement from the postsynaptic side. How those signals, pre and postsynaptic signals interact is another interesting, important question. And finally, universality and specificity. So I told you that SERP-alpha and JAK2 are critical for colossal connection refinement. Are they also used in other brain regions? And indeed, we found that JAK2 also regulates eye-specific segregation. In that sense, JAK2 serves as an elimination signal in multiple brain regions. At the same time, we believe there could be more specific signals, like during synapse formation, where there are multiple molecules cooperate to form specific synapses. We believe there might be specific signals to refine specific synapses so that refinement will happen precisely in the brain. So those are some of the questions, among others, remaining to address to fully understand what's happening in the brain during synapse refinement. So finally, the last slide. So these are the people contributed in my lab to the work I showed you today, critical collaborators, and the support. So thank you very much for your attention, and I can take some questions.
Thank you so much for that great talk. So please feel free to type your questions in the chat box or raise your hand. Larry?
Great talk and great graphics. So Hisashi, I remember at one time, maybe I have this wrong, but I think you had some thoughts about possible role of CNTF. And of course, the involvement of JAK, one thinks about members of the LIF IL-6 family as activators of that pathway. So what's the current thinking about that?
I think that's a possibility, absolutely a possibility. So indeed, so at least what we know is that GP130,
yeah,
which is a receptor that can activate JAK2,
yeah,
and again that binds IL-6 and so on,
yeah,
is partially involved. Meaning, if we inactivate GP130, indeed that affects partially the process of synapse elimination, but not fully. Meaning, so those factors are involved, likely to be involved, but there might be other signals that are contributing to synapse elimination. So that's absolutely one of the most important, interesting questions that we absolutely wanted to know.
Okay, thanks. Great talk.
Thank you. So we have a ton of questions in the chat box. So one of them is, does the proteome of the synaptic cleft have a direct effect on microglia about the synapse elimination decision?
I guess that's probably can be answered by Beth Stevens, maybe after her talk on microglia. But that's, yeah, absolutely.
Yeah,
Yes.
wait for that one. Yeah. So let's see. There's a bunch of questions. So are there any sex differences that are known in these signaling mechanisms during development?
So great question. So we do not know, but that's absolutely great question that we should address.
Another question, do we know if these pathways are also involved with inhibitory synapse refinement?
Another great question. So again, we do not know. But what we think is that the refinement of excitatory synapses versus inhibitory synapses could be a little different because in terms of inhibitory synapses wise, they also need to control the balance between excitation and inhibition. So meaning like they also need to sense the the inputs to the neuron when do perform refinement. So excitatory synapse refinement can be more local, because that's based on the competition. But then when it comes to the inhibitory synaptic refinement, I think that they also need to calculate the overall input to a neuron to control the balance between excitation and inhibition. So we think that there is some like a different mechanism involved. That's also indeed suggested by work by Binky and so on. But that's absolutely a great question, and we think there might be some differences, but we still need to know the mechanisms underlying the refinement of inhibitory synapses. Yeah, great question.
Thank you. So another question. I believe that JAK2 inhibitors are used clinically. Is there any evidence that they cross the blood-brain barrier or affect synaptic connections?
So, JAK2 inhibitors are indeed used or being under trial, meaning that it's kind of phase 3, I believe, for certain brain disorders. But that's partly because that suppresses inflammation. Meaning I think that's crossing blood-brain barrier, some of them. We do not know whether that also affects the synaptic refinement and so on, but that's absolutely a possibility to use for the treatment of disorders.
Thank you. Well, we have a couple other questions, but I want to leave time to move on to the next talk. So if you have press questions, if you can hold them until the general question and answer session at the end, we can get started with Dr. Stevens' talk.
Good, thank you.
Thank you very much.
Let's see. Okay, can you see my slides?
Yes.
Okay, great. All right, fantastic. Great talk, Hisashi, and perfect setup for my talk in a couple of ways. There's a couple of themes that you're going to hear come up again in my talk, but I'm going to tell you the story through a different lens, a different perspective, which is the non-neuronal perspective of pruning. And in particular, the themes that you're going to hear about, again, are activity-dependent competition. You're going to hear about specificity and molecules that are instructive and enable the elimination of specific connections versus others. And you're also going to hear a bit about how understanding some of these non-neuronal or immune-related mechanisms are helping us to understand mechanisms of pathological or aberrant synapse loss in a couple of different brain disorders, both developmental and in the aged brain, like Alzheimer's disease. So I'm going to focus a bit because you already got the big picture background from Hisashi, but I want to just kind of get you thinking about the non-neuronal cells of the brain, which is, of course, if you look in the brain, there are a lot of glial cells in there interacting with neurons and with one another. This is obviously a highly oversimplified cartoon, but I want to use it to illustrate a couple of important points. That one, that everywhere you see neurons talking to one another, and every synapse that you image or study functionally, they're in some way signaling to glia. Both astrocytes, which is sheath synapses and vessels, also even oligodendrocytes, and some forms of those cells can also synapse with neurons. And today I'm going to focus a lot, in the interest of time, on this cell, which is microglia, or resident immune cells. And I'm going to tell you a bit about how these cells, microglia, are pruning synapses and how they're doing that. Some examples using the visual system as a model system. But I definitely, even though I'm going to be more narrow than Hisashi did, and that I'm going to focus on this cell and some of these mechanisms, I wanted to just remind you that there's a lot of other interactions going on, bidirectional communication with other glial cells that I'm not going to talk about, but will obviously be part of the bigger picture as well. But I'm going to zone in on this cell here. And so I just wanted to say, you know, these are cells that have been studied for many, many years in my microglia, which are resident macrophages, brain macrophages and immune cells in our brain. They represent about 7 to 10% of the cells in our brains. And more, we're realizing that in addition to their established roles in inflammation and reacting to injury and disease, where they've been best studied over the last many decades, we now, through new tools like imaging and other tools that we have to manipulate and study them, are starting to appreciate these cells have a lot of normal physiological roles in the brain, especially during development when the brain is wiring up, which is how we got interested in them. And these cells, if you do 2-photon imaging, as I'm showing you here, you can actually watch these cells. This is just a loop just to show you how dynamic their processes are. But this has been observed by many, many researchers over the years. Not only do they sort of survey the brain parenchyma, including synapses, but they're also incredibly responsive to local perturbations or changes in the environment, including synaptic activity. This is an extreme example where you can make a laser ablation and you can see this like dramatic response. This is like an injury paradigm. But even under normal conditions, these cells are just really receptive to neuronal and even synaptic signals. They have a host of receptors for neurotransmitters and for other growth factor related cytokine receptors that they just are really well positioned to be listening to what's happening in the brain. So I'm going to hone in on one function today based on the topic, which is synaptic pruning. But I wanted to say they do a lot of other things in the brain. They exist in a lot of different states that we're beginning to study more carefully. But I want to do a little bit deeper dive on their role in synaptic pruning. So as you heard from Hisashi, so I don't need to get into, really this question of synaptic remodeling or synaptic pruning is happening in different critical periods at different times and places in the brain, certainly not an all or nothing process. But what I've been always intrigued by is the fact that because once these critical periods are defined, you know, it raises all kinds of interesting questions about whether the same mechanisms that might help remodel synapses early in development, let's say in the sensory system, like the visual system, are they the same cues and mechanisms that also refine cortical circuits or prefrontal circuits later in development? We don't know, but I think using an approach of a model system that you can deeply study, like for example, the visual system has enabled us to pick a circuit in which we know a lot about pruning and start to really dig in deeply about understanding not only the mechanisms, but the role of these immune and glial cells in the process. And I also just want to say that understanding synapse loss, which is also a hallmark of a lot of disorders, the idea that although all these disorders are quite different, there could be some common biology or common mechanisms that might be relevant to all of them to make synapses vulnerable in these diseases. And that's one of the overarching questions that we've been interested in as well. But I think the more and more the field is starting to appreciate there's some conserved mechanisms across development and disease. And more emerging genetics are telling us that some of the mechanisms that we're studying in development are also relevant to disease. In the case of the immune pathways I'll talk about, microglia have been implicated in Alzheimer's and many other disorders, and that's more and more seems to be the case in other disorders as well. So going back to the kind of cartoon that you just heard about, this idea of activity-dependent synaptic competition and synaptic refinement, again, this sort of illustrates on the right sort of two red and blue inputs that are competing for synaptic territory. This happens to be a cartoon of the retina geniculate or the mouse visual system, but it could be in other areas as well. And the idea that the less active are the ones less able to fire the postsynaptic cell more efficiently tends to be the ones that get eliminated on the presynaptic side. And so we wondered, could the microglia, which are associating with synapses, especially during these developmental windows, could they be remodeling synapses? That was the question that we addressed a number of years ago. And as you can see here, we observed their processes interacting with both post and presynaptic elements in different contexts. This has been shown by many now over the years. But just looking at them, seeing them touching synapses doesn't really tell you that they're actually pruning synapses. And it's been hard to catch them in the act. So we decided instead to go to the retina geniculate system, which is this well-established model system for studying synapse elimination, thanks to the pioneering work of Chen-Fei Chen, Carla Schatz, and many others at Harvard that had really pioneered this model system to study mechanisms. They've been studying it on the neuronal side, and so we decided, okay, let's look at the glial cells in this process. So you can label the retina with anterograde tracers of 2 colors, which can label the projections into the visual thalamus or the DLGN. And we know they segregate into these eye-specific territories. So if you were to look at this postsynaptic relay neuron, it might be looking like this early on in development, intermingled or innervated by inputs from left and right eye. But by the end of the critical period, which in a mouse is about P10-ish, they're mostly segregated into one eye or the other. So the elimination of the inappropriate eye and the strengthening of the appropriate eye. So again, this has been well established. Now we wondered, could the microglia, one, are they actually engulfing or remodeling these inputs because there are resident phagocytes in the brain? They have a lot of phagocytic receptors, and we observed they were more phagocytic during this period. And more importantly, are they actually doing this in a more sort of specific manner? Are they receiving instructive activity dependent cues? And this is still an active question that we haven't completely nailed, but we have evidence to support that they're not just cleaning up, they're not just the garbage collectors, but they're actually responding to activity dependent neuronal cues. And I'm going to tell you about a couple of those today. But this was the model system where, again, zooming in on the circuit, you can label the inputs. And Dory Schaefer in my lab a number of years ago, using a transgenic line where the microglia are green and high resolution imaging, this is in fixed tissue, not live imaging, essentially showed that during this window of refinement in the LGN, microglia were engulfing or phagocytosing into their presynaptic, they're basically the presynaptic elements were found inside the lysosomes of the microglia during this window of pruning. And importantly, when we drove activity dependent competition, so basically you can make one input more active or silent with tetrodotoxin, you can drive competition. No matter how we did the experiment, they were more preferentially engulfing the less active eye input. So that was an important finding because it said that these microglia do care about activity and that they had a preference for the less active eye input. And also this pruning wasn't happening all the time. It was happening in these little windows of development more than others. And we went on and showed that one of the many ways actually they recognize synapses and eliminate them through this phagocytosis mechanism is through this group of innate immune molecules called complement. These are secreted immune molecules that have been very well studied in the immune system. But when I was a postdoc in Ben Barris's lab, we showed that a lot of these complement proteins are expressed and localized to synapses by high resolution imaging, for example, throughout in the brain, but also especially during developmental critical periods. And that microglia, which express receptors that recognize these secreted proteins, have the ability to then phagocytose these tagged synapses. So somehow these secreted molecules are targeting or tagging presynaptic elements, but also in some cases post. And that was what we went on to show, that if you blocked either various complement proteins or the receptors, that lead to defects in synaptic pruning and connectivity by anatomy and by physiology. So this is that eye-specific segregation. And if we did those eye-specific segregation experiments in the knockouts, there was a lot more overlap going on. And by physiology, which is using the Chen-Fei Chen acute LGN slice prep, we could also show that the knockouts were maintained some of this extra synapses. So they were not refining properly by electrophysiology. And the two together show complement as one of the ways this works. And so that, of course, introduces a number of questions you're probably wondering, like, how is it that these secreted immune molecules are doing something as specific as synapse elimination? How do you get specificity, right? So we showed some evidence that there was preference. We showed evidence also that complement was important, but how does it actually work? And this is still something we're actively working on in the lab, but I want to tell you about the model. And also, not surprisingly, it's never about just one set of molecules, as you just heard from Hisashi's talk. And so one model is that during this activity-dependent competition, these eat me signals, which are what complement is in the immune system, these are the same proteins that would tell a macrophage to remove an apoptotic cell or a pathogen, that somehow they're landing on presynaptic elements or presynaptic terminals, and that might be recruiting microglia to those synapses, those less active synapses. but it doesn't explain why the red ones don't get pruned, or why, for that matter, the whole axon or the whole cell doesn't get pruned. So what's stopping them from nibbling all the way up the axon? So then we went back and started thinking about how macrophages do this in the immune system. And we realized that a lot of these immune molecules that we kept calling immune were normally expressed in the brain, both receptors and the ligands. And there's a whole group of molecules, complement inhibitors, for example, and molecules that are called don't eat me signals that prevent a macrophage from engulfing a healthy cell or a self cell, for example. And as we started really looking at that, it turned out there's a whole bunch of these molecules in the brain, especially, as you just heard from Hisashi, CD47, which is expressed in neurons and continues to go up and localized to neurons throughout brain development and into the adult, localizes to synapses. And you heard from Hisashi about SIRP alpha, which is obviously also an important synapse elimination molecule on the neuronal side that gets cleaved. But microglia also express SIRP alpha, especially in these developmental windows. There's a lot of SIRP alpha on the surface of microglia. And essentially the proposal was that maybe it's these don't eat me signals that are preventing microglia from engulfing synapses. And indeed, we went on to show if you knock out CD47 or SIRP alpha, we could show that microglia over prune. So they engulf too many synapses. It's the opposite phenotype. And moreover, that led to enhanced refinement. And also we found, not shown here, this was also activity dependent, in that if we did that same competition assay I told you about before, microglia no longer know which one's weak and strong. They don't have a preference anymore. So we're still trying to understand exactly how this works, but we think maybe what's happening is there's a combination of cues. And when those surveying microglia, which are surveying the brain parenchyma, they express a lot more receptors in this, but these are the two I told you about today. If they basically survey around and they encounter different combinations of cues, it's these instructive cues on the synapses that tell the microglia, engulf the synapse that has complement, the eat me signal, but lacks CD47. And we think that might be one way you get specificity, and exactly that's how it works in the immune system. So I think it's a great example of a borrowed mechanism that's working in the brain. And we've shown some data that supports this model so far. But it's a lot of other questions. What are these secreted molecules binding to its synapses? What are the receptors, if you will, for complement? And work I'm not going to have time to tell you about today, but was just published recently in this past year. Scott Hewitt in my lab showed that another immune or sort of more conserved mechanism, phosphatidylserine, which normally is in the inner leaflet of a healthy cell, and when there's apoptosis, it flips to the outside, and is a known eat me signal. And what Nicole found is that this phosphatidylserine is locally exposed or flipped in certain synapses during development. And that's one way that microglia recognize and complement gets recruited to synapses. We also are really interested in time-lapse imaging this to get at that chicken and the egg question, and developing a lot of tools based on some of this work I just told you about today, so that we can get more clarity on the timing and the dynamics of this process. So there's lots of work still in progress. But in the last bit, I want to try to seg over. Before I do that, I told you I wouldn't completely ignore the other glia. I wanted to highlight that astrocytes, remember that these are the other glial cells that are really important in a lot of brain functions and development, including synaptogenesis. But work in Ben Barris's lab, as well as Mark Freeman's lab, has shown that astrocytes also phagocytose synapses in the developing visual system in an activity dependent manner. But interestingly, they don't care about complement. They do it through different mechanisms. These phagocytic receptors, MEGF10 and MERCHI-K, which are really important in terms of clearing cells and debris, they are actually how astrocytes recognize synapse elimination. And so this is just a good example of how the different cells can prune and even engulf synaptic elements in the same system, but they do so by different cues. And just to kind of generally say that more and more data is coming out of so many labs that not only are different glial cells like microglia and astrocytes doing pruning, but there's also a whole bunch of other immune-related molecules expressed in the developing healthy brain, including work by Carla Schatz that's shown the MHC, which is part of the innate adaptive immune system, also important in pruning. And so the more we learn, the more we realize that the brain, much like the immune system, is very complex with respect to these different molecules. And even though the complexity is a little bit overwhelming, I think it is in the same way that it's very specific signaling happens in the immune system. These same molecular mechanisms are what gives the immune cells or the glial cells those instructive cues. And I think it actually makes a lot of sense. So the last bit I just want to seg over before I turn it open to questions is I just want to end with what I mentioned at the beginning, which is that we've uncovered, and now others have uncovered, that some of these same immune and glial mechanisms that regulate synapse elimination during development can become aberrantly activated in the adult brain or in the diseased brain if the brakes go off. So this idea of developmental pruning could be disrupted if these immune pathways like complement are dysregulated. And I want to give you a couple of quick examples. They're published work, but I just want to paint the stage with the point that if we didn't really understand the normal mechanism, I don't think a lot of the disease relevant mechanisms would make a lot of sense to us. But I'm going to focus just on two examples. One is some evidence in Alzheimer's disease, where we now have genetic evidence that implicates both complement and microglia. And I'll tell you some evidence that supports this model. But also developmentally, schizophrenia, which is a neuropsychiatric disorder, is also some evidence that also links the complement system and the immune pathways I mentioned in synapse elimination in that disorder as well. Now, it's been long studied and hypothesized that aberrant synapse loss in the context of schizophrenia might underlie some of the defects, and especially cognitive relevant defects in schizophrenia, which is a neuropsychiatric disorder. And there's a lot of pathological, so postmortem evidence that suggests there's spine and synapse loss, especially in the prefrontal cortex. And imaging studies also suggest that might be true, but it's been really hard to get at in terms of cause versus consequence. There's a lot of things that can cause synapse loss. But emerging genetics over the last 5 years or so are starting to directly implicate some of the same pathways I just told you about, including the classical complement cascade, and another molecule called CSMD1, which is now a putative complement inhibitor. And this is really work that was really an important study where Steve McCarroll, HMS colleague and my collaborator and his graduate student, excitingly discovered that in the genetics of schizophrenia, where there's been a lot of GWAS studies that uncovered a lot of signal, the strongest peak in the Manhattan plot of association signals is in this MHC locus, which of course is a locus that's been very mysterious because it contains 100 or more genes or variants that could be potentially regulating that signal. And long story short, what Ashwin, who's an MD PhD student in our neuroscience program, discovered, they fine mapped this strong signal, almost the entire signal, to the complement C4 gene. And basically the take-home message is that individuals can express many copies of A or C4B. There are two different genes in the human. And what Ashwin discovered was that individuals that express a lot of C4A, but not B, have significantly more risk of schizophrenia, and they have more C4 expression in the brain. And this nominated a lot of potential mechanisms by which maybe, if you put that together with what I just told you, maybe the way that's working is that it's leading to too much pruning. And this is ongoing work, collaborations between Bernardo Sabatini, Mike Carroll, Steve McCarroll, and many others in our Conti Center, where we're now testing the hypothesis that maybe too much C4 means too much complement-mediated pruning. And of course, that can't explain all of schizophrenia, but it's a mechanistic hypothesis that we're actively testing. And indeed, we have evidence for too much C4 at neurons and synapses, both in the human brain and human neurons. We have evidence published that C4 knockout mice also fail to prune in the visual system, much like the C1q and C3 knockout mice. And now, thanks to new tools, we have new evidence that suggests a humanized mouse model that expressed too much of that C4a, the risk variant, leads to excessive pruning. So this is work in progress, but the model might be that making too much C4, either because you lack the brake or that you have too much C4, could lead to an over pruning of vulnerable synapses. And now work ongoing is aimed to test this hypothesis using a combination of humanized mouse models, IPS models, and ultimately going back in the human brain to test this. And the other thing that we're doing is asking whether pruning could help explain the age of onset of schizophrenia, because that usually happens in adolescence or early adulthood, which in the prefrontal cortex, this is the last area of the brain to really mature and refine and prune. And so we wonder, could this explain why the timing of schizophrenia is so late? So using a combination of these different animal models and including non-human primate models, we're now leveraging the genetics to manipulate these pathways to test the behavioral and the functional consequences to see whether this hypothesis may actually fit with this idea. And in the last couple of minutes, just say that in addition, work from a number of groups now have shown that this complement-mediated pruning mechanism is relevant to other disorders as well. And in particular, Alzheimer's disease, which there's a lot of evidence in human and animal models that there's an early synaptic loss, much before you start to get the cognitive and pathological hallmarks of Alzheimer's. And using a similar approach, using these pruning assays and complement deposition assays, work by So-Yeon Hong in collaboration with Dennis Selko and others have shown that these complement molecules are not only upregulated, but they're sort of targeting these vulnerable circuits and synapses. And that if we knock them out genetically or with an antibody, we can protect the synapse loss in a number of different animal models. And this is just shown here. We can also block it with a functional antibody. And so it's kind of painting the picture that, okay, maybe this is a kind of downstream common converging mechanism, but timing and context matters. And I told you a lot about synapse elimination mechanisms, but we're also appreciating that microglia are implicated in Alzheimer's, for example, and other disorders, and there could be other mechanisms at play as well. So we're kind of combining the sort of functional basic science mechanisms to also more deeply characterize what other mechanisms might regulate this independent of complement. And also, how do we better define and study microglia activation and function in the context of not only animal models, but also in the human. So a lot of single cell sequencing is starting to happen in the field is pretty exciting. So the idea that we might be able to come up with molecular markers that denote a microglia that shifts from this homeostatic surveying microglia to one that's excessively pruning synapses, or one that's potentially beneficially clearing plaques or more inflammatory. And so work ongoing by many groups in the field now are starting to use single cell transcriptomics, proteomics, and epigenetic studies to more deeply characterize the different states of microglia, with the hope that maybe we can figure out a way to manipulate just that aberrant state, the bad state of microglia, and figure out ways then, based on the basic science, to target just those particular mechanisms and pathways. This is just to show the first of these studies was by Ito Amit's group. But ultimately, we're kind of trying to bring all this together, leveraging the genetics that have given us clues about the pathways. And this is in Alzheimer's, but this could be relevant to other disorders as well, to understand better how to link the genetics with different states, transcriptomic and otherwise, to specific functions. Well, we think that might enable us to identify and track disease relevant pathways, including microglia, as well as new biomarkers. So I'll end by saying there's a lot going on with glia. So when you think about circuits and you think about the brain and health and disease, the glia are active participants, and they're if something happens to the neurons, whether they're the primary insult or not, it can influence the glia. And vice versa. And so the idea of kind of bringing these ideas together and studying them together, not separately, is what I think the field is moving towards. And Hisashi and I have a number of collaborations that we're doing together that I think illustrate that really beautifully, where Hisashi's been studying SIRP alpha at CD47 and through the mechanisms he shared with you on the first part. We'd been studying this in the microglia part, and now how do these come together? And we have lots of experiments that we're already planning to do together along those lines. So I'll just end by thanking the human microglia, who not only are an amazing group, but they also brew a fabulous beer called Microglia Ale. So we started with beer. I didn't know you were going to put the beer thing in there. And I'm going to end with beer in my last slide. So thanks.
Thank you so much. That was a wonderful talk. So yes, let's take some questions that are specific to Beth's talk, and then we can move on to general Q&A. Okay, we have one in the chat box. Let's see. Does the lack or absence of dendritic spines affect the synapse pruning, or can it not support synapse function? Let's see.
Sorry, can you repeat that one?
Yes. So I think the question is asking about does the lack or absence of dendritic spines affect how the synapse pruning occurs?
Does the lack of spines? Sorry.
Yeah, the lack or absence of dendritic spines
Okay, I'm not sure exactly what the question's exactly asking. I don't want to misinterpret it, but I did want to say that I talked a lot about presynaptic pruning today, like a lot of things on the axon side. And I think it just reminds me to say that I gave you kind of, the presynaptic side of the story. I did not tell you about spines and postsynaptic side. And in the same way that Hisashi brought this up at the end, there's clearly a postsynaptic signaling going on that is potentially also activating and regulating this process, immune and microglial. And similarly, in other contexts and other groups have shown that microglia engulf spines and and eliminate postsynaptic elements as well, perhaps through different mechanisms. So in the same way that there could be different molecular cues that regulate inhibitory versus excitatory synapses, I think there's also going to be different molecules and mechanisms that regulate pre and postsynaptic remodeling, let's say by microglia, depending on the timing, the context, and the signals, right? So, so that's a long-winded answer, but I think I did want to make sure I didn't suggest that all of this was presynaptic. There's evidence for postsynaptic as well.
Thank you. So I encourage people to raise their hands so that you can clarify your question, if I'm not reading it correctly or you want to explain further. But let's move on. So there's some other questions. So is there direct evidence that neural activation can increase the don't eat me signals?
Okay, great question. We're studying that actually. So the work I told you about, we did have some evidence that the localization of that CD47 molecule does change when you manipulate activity, and more particularly when you drive synaptic competition. So we are also interested in the idea that maybe activity is upregulating or downregulating these molecules, and this is active work in the lab. But the other thing I think is as important as expression, like mRNA expression, is the localization of these molecules. And so in that activity-dependent competition model in the visual system, for example, CD47, which is normally like all around on the membrane of the synapse and the axon, we found a redistribution so that when you look at less versus more active input from the two eyes, we did see a shift, which suggests there's got to be some signals that's regulating not just expression, but localization. And I think for microglia and glial cells, the localization is really important. It's that combination and where they sit on the cell or the synapse that's ultimately directing them to those particular structures. It's a great question. And we're designing now assays to image this, as I mentioned, so we can get a better handle on like exactly what the dynamics are and what's the kinetics of this process. So there's still a lot we don't know, but it's a very good question. Thank you for that.
Thank you. So another question is, what do you think about the controversy of whether microglia in mice reflect human signaling processes?
I don't know if it's so much of a controversy yet because we don't know. I mean, I think that what's slightly controversial is a lot of like sequencing going on and comparing human data from mouse and human, and there's certainly going to be some things conserved and some things that are not. But I think part of this is like we're early days and there's a lot of reasons why there may be differences. And there is a lot of conservation, both in some of the same mechanisms and molecules and even by single cell, some of the same states. But not surprisingly, of course, not everything's going to be conserved. So I think that's part of where all of this more broad effort in the field to start to really bring together multi-species data sets using a lot of different approaches to try to better establish what are those pathways that are conserved versus not. And I think that's where we're kind of letting the human genetics guide us a bit, because if the genetics are nominating some of these pathways like TREM2 and CD33 and complement, that's telling us there must be something there to study more deeply in the mouse. But there may be some things that are more relevant to the model itself. And so I think we're going to start to try to disentangle that as the data comes in.
Thanks. So another question is, are these targeted sculpting mechanisms uncoupled from generalized inflammation, meaning ongoing non-acute inflammatory drive, poly-IC? Could that derail a targeted pruning mechanism, or are they too tightly regulated?
It's a fantastic question. I think a couple of things to say about that. I mean, the word neuroinflammation is also kind of a loaded word. It means a lot of things depending, again, on the context and what you're asking. But if you put it in the context of like what happens when there is a lot of inflammatory signals in the brain, that could be aging, Alzheimer's, a situation where there's an injury or a challenge. One thing that is most likely happening is that there's a dysregulation of that neuroimmune signaling in microglia, let's say, so that maybe the cues that normally would give them that specificity, where they're more kind of physiologically able to do their day jobs the way I just described, if now the inflammation is outweighing their normal physiological functions, that could shut down or dysregulate their ability to do that. And I think that's part of the challenge now, is asking how, under different specific challenges relevant to disease or injury, how does that influence their biology and their specific functions like pruning? And that's exactly the kind of questions we can start to ask, because we can do the same kind of experiments I told you about today with and without a challenge, and then ask what happens to the molecules, what happens to the mechanisms. And so it's a work in progress. And we're also doing that in the context of that schizophrenia work I mentioned, because there we know there's genetics, like kind of leading us towards a pathway. And that means over pruning potentially. So now we can say, how do genetics and environmental challenges converge to regulate this process of pruning? And so we're doing that again, using those animal models by taking advantage of the ability to overexpress the human variant C4, and then at the same time, add a challenge like inflammation or stress, and then ask what's the outcome of that. So we don't know exactly how it works, but we suspect that the normal mechanisms are going to become dysregulated as a result of inflammatory signaling.
Thank you. So maybe we'll do one more specific question, just open up the discussion after that. So is there any data that there are differences between microglia across brain regions? I ask because there is region specificity on where synapse loss occurs in neurodegenerative disease.
Excellent question. One we're very interested in. So a lot of the first pass studies on single cell, which is very new technique over the last few years, a lot of those studies have been taking the whole brain, or in some cases even purifying the microglia out of these models. Now what's clear, since we know more about how to do this and what to expect, we can start to look in different brain regions: hippocampus, frontal cortex, cerebellum, using epigenetic assays and single cell and others. There's clearly differences, region-specific differences, probably not surprisingly, because the environment in those brain regions are going to be different. And so this is still, work that's ongoing. But I think the idea that why is there vulnerability in certain brain regions and circuits is a big, big question. So we want to better understand, why or if these immune mechanisms, including microglia, are different in those regions versus others. And maybe that is underlying some of that vulnerability. So don't know completely the answer, except that there's emerging evidence that does suggest that there are region-specific differences. And these cells, microglia, are so incredibly dependent and responsive to environment. And so I think that, but they're also, their intrinsic states are going to maybe also matter as well. So again, trying to disentangle their cell intrinsic mechanisms that might make them different with the environmental signals. And that can only be done by doing a real systematic analysis across different brain regions in mouse, human, non-human primate, other organisms, to see how that comes together.
Thank you. So we have more questions in the chat, but I also wanted to just take advantage of this time to open it up for questions that are both for Dr. Stevens and Dr. Umemori. So if people would like to raise their hand. So yes, we have a question from Mitsuko.
Hi, nice talk. Thank you so much. So the very massive pruning happened during development and also in disease state. But as Beth told at the end, the same machinery is used in the adult with disease state. So that means the machinery stays in adult. So do you think the more finer pruning in adult can also use the same machinery that today you have presented in today's talk, or it's only during the development and also in disease state?
I'll let Hisashi, why don't you grab that first, and then I can chime in if you want.
So I guess at least during development, there's a robust refinement. So clearly, those molecules, neurons are more plastic, and pruning happens in a more robust way, and those molecules are used for refinement. So I guess in normal adults, I guess those molecules are kind of stabilized, meaning after the critical period, neurons are more stabilized, so that those pruning or activation of those molecules wouldn't really happen, so that we can keep a more firm connections. But for some reason, probably during the disease development, those stabilized systems would be somewhat become more plastic again, so that those molecules or mechanisms can be reactivated to have a synaptic loss that causes disorders. So I guess systems are there, but in adult brains, networks are protected. That's why normally we do not develop disorders. But something goes wrong to cause the brain to be more plastic, then that can lead to disorders. That's I think maybe what I would say what might be happening during development as opposed to disorders.
So it's not known that it's used for the normal learning or
like more synaptic plasticity, like just more sort of more local, like let's say thinking about that kind of remodeling versus like the developmental removal of structures completely. You're thinking maybe more like plasticity of synapses?
Okay, yeah. Then yes, yes. The answer is yes. Those molecules are indeed used for plasticity. Like molecules I just talked about, for example, JAK2 is involved in LTP and LTD, more like LTD, and SIRP-alpha as well. Like if you knock down those molecules, indeed those will affect LTP or LTD. So during plasticity, indeed those molecules used. In that sense, not just during development, but in adult, activity dependent processes also involve those mechanisms. Yeah.
Yeah. It's a great question.
Thank you.
We have another question from Oystein. Sorry if I'm mispronouncing your name.
No problem. Hey guys, great talk. So I was basically wondering, is the complement cascade self-aware in a sense? So if you start to mess with it, for instance, potentially trying to block some of these runaway pruning mechanisms, and you try to reduce C3, for instance, does that make the complement cascade very angry? And then it can how careful do you have to be when you mess with the with this cascade?
Yeah, so it's a question that's really an important one to answer, because if you were going to start thinking about therapeutically manipulating complement, right? What are the impact of doing that, right? And so we've been thinking about that because complement has a lot of other important roles in the brain beyond pruning. I told you just about one today, but depending on where you block in the pathway, it can have very different consequences. So when we tried this study that I mentioned earlier, the Alzheimer's study as an example, where this was the work in collaboration with Dennis Selko and Cindy Lemire, where we took two approaches. One was genetic, where we knocked out C3 or C1q, and that protected the synapses. Another was a C1q blocking antibody that only manipulated the activation of the cascade. And we saw similar protection, but the idea that C1q, which could be more therapeutic way to do this, it wouldn't shut down all the rest of the cascade, which is why they chose to go after C1q. So there's other arms of the cascade that converge on C3, and then you can still get that role. So because all the data so far supported C1q, C2, C4, C3, that's one way to manipulate just that part without affecting the other functions. And, I think it's still a big question, though, is what happens if you shut it down? So you have to be really selective with how you do it. So thinking about it from how to get even more specific, if we could identify the complement binding receptor at weak or vulnerable synapses, then you could develop a small molecule after that, but it wouldn't affect peripheral complement. It wouldn't affect all the wonderful things it does, like clear plaques, for example. So I think that's just illustrates more deep mechanistic insight, gives you more ability to target the pathway that's detrimental and leave alone the beneficial pathways.
Thank you so much.
You're welcome. I just also wanted to add that thinking about Alzheimer's and age-related diseases, trying to target that, maybe you weigh out the cost-benefit analysis of manipulating complement, but in development, like schizophrenia, it would be really tricky, I think, to start mucking around with complement or some of these pruning pathways during a period where there could still be a lot of remodeling in the brain. So I think kind of manipulating it in development would be a bigger challenge. So it's something else to think about.
Yeah, that's why I was asking about the poly-IC and things like that. You know, like if you have a sterile or like a unresolved complement activation, like the great Yin paper, I think Nature Medicine last year, they had, you know, ApoE, lipid deposits, complements. I was thinking, you know, so like if you target it to try to mitigate, for instance, aberrant pruning, but then there is base drive in other cell types, and you kind of miss those microglia, and maybe even hit some peripheral cells if you give an IP injection of something with blood-brain barrier permeability, and suddenly these peripheral cells get enraged, and then they just ravage you outside of the brain, you know. But like, yeah, because sometimes it's hard to know if you have a residual lingering infection, like for instance, post-COVID, certain lot of neurological symptoms, even a case of sudden onset PD, I believe. So that's things like that I was thinking about.
Yep. I think that you've raised a more general challenge with this neuroimmune work, right? Is so many, like if you want to target microglia, well, how do you do that without affecting peripheral macrophages or the ones in the borders? Like you have to get specificity, which is why that single cell I think is going to be powerful, because you could try to get more specific at which population you might want to target. And then similarly with the complement cascade, don't go after the whole cascade, go after the specific mechanism. And I think that's the way we're thinking about it. And maybe that's in general thinking how to kind of get around some of these other issues that you raised.
Absolutely. Thank you so much.
Thank you. Derek?
Yeah, hi. Great talk today. I was wondering, is there anything known about microglia astrocyte crosstalk, and if they can influence the other's ability to engulf synapses?
Yes, there is. There's a lot of interest in this in the field now. A lot more work actively going on to probe the astrocyte microglia communication. Actually, Frank Matana and others at Harvard are working on some of these questions as well. But the answer is yes. I think the two are like, you know, that image that I showed was just overly simplified of astrocytes and microglia. I mean, they're literally all right there talking to one another. So if you influence one, you're probably going to influence the other, both in development and disease. For trying to dissect out like what are the key signaling between the astrocyte and the microglia, receptor ligand interactions, and how that can influence the synapse is something that is of huge importance. The problem and challenge with that is you can't really take all these cells out of the dish and put them in a dish because they really change state. So you have to study them in vivo, which means you have to be able to figure out what to manipulate to study it. And I think that's where a lot of the omics studies are being helpful. Because if you do this really thoroughly, that can nominate receptor ligand interactions that you might target on one cell versus the other. But in a summary, I would say there's a lot more data suggesting that if you block microglia's ability to prune, let's say in that Ben Barris's paper I mentioned on the MERTK, they blocked MERTK and astrocyte pruning, and then the microglia pruning kind of took over. You might imagine they kind of work together, and when one can't do it, the other one steps in through different mechanisms.
So I have a big picture question for both of you. I know that both of you have an interest in disease and in addition to basic science, and one of the purposes of this series is to promote crosstalk between those kinds of research. So it seems like human genetic data, such as the schizophrenia research with C4, as well as like looking at synapse loss, things like that, the human research really drives what you do in basic labs with mouse models, etc. And I was just wondering, are there if you had a wish list of experiments, a clinician could do for you, what are things that you're really curious to find out about, or a clinician or a human geneticist?
That's a great question. I'm going to turn it over to Hisashi first.
No, you go ahead.
Sure? Okay, because I mean,
you're doing human-based studies, so
yeah, I do have a wish list. I mean, I think what we're realizing now is like how we really want fresh, high-quality human samples from the right cohorts that enable us to test hypotheses, right? Which is really challenging. Like if you take the case of schizophrenia, like we don't have biomarkers for who's going to convert and have schizophrenia and psychosis versus someone who doesn't. So you don't really have a way of tracking. And obviously these are highly polygenic disorders where lots of genes contribute and other factors to risk, right? So if we had the ability to do more longitudinal analysis of not just brain, CSF, blood, paired samples from the same, I mean, I know that's a dream, but if you could do that, you could start to correlate over time or with and without different genetic background or relate that to cognitive and clinical symptoms. If you could do that really kind of rigorously with a large sample size across a lot of collaborative efforts, the idea might be that you start to really get through the noise and start to identify, mechanisms that you could also read out in the blood and CSF that are relevant to what's happening in the brain, right? And as an example, which is, of course, these are really hard to do, right? All those longitudinal studies, you might get blood, but you wouldn't get CSF and blood, or you wouldn't get the brain from the same person while they're alive. So we recently started collaborating with a neurosurgeon up in Finland who sees a large cohort of idiopathic normal hydrocephalus patients. And the bottom line is in these particular cohort, they do collect brain, blood, CSF from the same individuals, and some do and don't have amyloid and tau pathology. And this is just an example where through collaborative efforts and just saying, hey, this is what we really need, this neurosurgeon jumped in and said, oh, I have samples that could be helpful to you. And so we're starting in collaboration with Evan McCosco and many others to start to do kind of the deep dive analysis across all those 3 samples from the same patient that do and don't have amyloid and tau. That's not Alzheimer's, but what if we learn something that could then tell us about what to look for in those schizophrenia or Alzheimer's or other samples, right? So that's just an example of like partnerships with, surgeons and neurologists and clinicians that could help to collect the right samples the right way that can enable the basic scientists to ask specific questions.
Yeah, I agree. And I guess what we do not know is whether and what kind of like a synapse refinement or synaptic loss is indeed happening in the patients or disorders, like where in the brain like a particular synapse is lost or even during development, like a house, like a synapse refinement is happening in human brains, right? We still do not clearly know. So based on human samples, disease brains, if we know like more characters or specific changes in the brain, we hope the knowledge from basic science to be applied to treat disorders in a more specific ways. Like I talked of molecules, but the molecules are just some tools. But rather, we want to use those to indeed correct like a synaptic loss or like a diseased brain. So for that, we also need to understand more in detail about human brains and human patients. And those are something we really want to know from human brains of patients. And then that's something clinicians can also try to look at or to collaborate with us to fully understand what's happening in the human brains or diseased brains. So that's something I'm hoping.
Yeah. You just sparked another thing just to mention is like, imagine if we could image synapse loss in real time in patients non-invasively with a PET ligand, right? And right now there's some efforts in this field to try to start to do this. So there are microglial PET ligands that are not very specific, but they're useful in saying, okay, these cells are changing, but what if you could actually measure synapse loss and synaptic changes with something that's targeted to the synapse in some way? And so there's a development of this SV2 presynaptic PET ligand that's now being used in patients. It's not clear if it's going to have the resolution to really reveal synapse loss in, let's say, early Alzheimer's, but I would say that that's just an example of how if you had a tool like that, you could start to track things in addition to things like amyloid and tau and other biomarkers. You could start to track synaptic density changes. And I think it would be wonderful if there were ways to do more specific ways of getting at the synapses, right? And that's just one that's ongoing, but I think others are in development now.
That's wonderful. Really interesting to hear about. So next up is Victor.
I have a quick question for Beth Stevens, namely whether there are functional differences between microglia at different organism ages in terms of their ability to remove synapses.
Yep. So we're doing a lot of kind of deep profiling, both transcriptional and functional across development into adult and aging. and trying to really like connect the dots between the transcriptional signatures that are definitely different in adult versus young, and then how that reflects their function, right? So I think that they are quite different, especially because the brain environment is quite different. But then under certain conditions, they can be triggered to kind of kick back on some of the same pathways they had earlier. And one hypothesis that we're testing is whether, again, going back to the theme of Hisashi's lecture and the general theme that kind of threaded through mine, is how is neuronal activity maybe an instructive cue that might kind of reinvigorate or sort of reactivate these processes. And aberrant neuronal firing and aberrant activity is known to also happen, especially early in a lot of these disorders. So one idea might be it's this aberrant firing that's almost like kind of tricking the microglia to thinking, okay, well, we got to remove the synapse, or sort of these activity dependent cues kick back in. But the difference now is they're not supposed to prune those synapses, and then they're dysregulated. So again, the idea of upstream kind of targeting the aberrant activity is one of the things that might be, because these are plastic cells. So it could be, if you give them the same cues again, could they go back and do what they used to do? And we don't know for sure that's true, the answer to that, but that's one idea, one hypothesis. So they are pretty plastic.
Thank you.
I was looking through the chat questions, and there's one I think you might have touched upon, but I wanted to see if we wanted to talk more about it. Someone had asked, are there indications that the JAK2 SERP alpha axis that Hisashi spoke about could be involved in neurological disorders?
Yes. So yeah, the answer is yes. I mean, of course, we do not know whether like a defect in synaptic refinement is causing disorders, but like, those molecules are implicated in various neurological disorders, like schizophrenia, for example, and also like in adults, like Alzheimer disorders and so on. But that's an implication. But like, absolutely, those molecules are implicated in various disorders. Yeah.
I think we are out of time, but thank you both so much. Those were really fabulous talks and really fabulous discussion, and nice to hear, how your work can come together across labs. So yeah, thank you so much, and thank you to everyone for your active participation and questions.
Thanks for having us, and thanks for listening, everybody, and your great questions.
Yeah, thank you, everybody.
Thank you very much.
That was fun, Hisashi.
Yeah, thanks.
Now we got to go do the experiments.
Yeah. Yes. Great.
Bye.
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 Bye now.